Degree. And Dr. Soundararajan, Assistant Professor from the Department of Physiology, Environment Institute of Sciences, located in the Bharat Institute of Education. The topic that we are going to discuss today will be the introduction to endocrine physiology mechanism of hormone. As you can see in this picture gives all the list of endocrine. We have the pineal gland, pituitary gland, thyroid gland, also the parathyroid glands. We have thymus, adrenal gland, pancreas, the endocrine pancreas, the ovaries and fingers, testes. These are the endocrine organs in our body. What basically is a hormone? A hormone is defined as specialized organic that are synthesized with endocrine tissues in response to a specific stimuli and exert their precise effects particular organs. So we will take this definition. What are these hormones? These are specialized organic molecules. Now, what is the stimulus for all these things? There are going to be specific. What's the definition? Endocrine hormones. And what are they going to do? They are going to target specific cells and they are going to be there. What are the next thing that we are going to do is see hormone actions. This hormone accomplishes the binding receptor as a target tissues. So every hormone binds to a receptor the target tissues. Now, what happens next? Activation of these receptors leads to alteration in intracellular receptor signaling pathways that changes the cell function. What is the meaning of this? The hormone basically activation. They go and find some receptors that are the target tissues. Once these receptors are activated, then there's going to be a signal inside the cell, which is going to be different signal. There are different signaling pathways that is going to change cells. This is a simpler picture which gives what I just told in the last time. There is a first messenger here. This first messenger is actually the hormone of the hormone. This is going to act on the receptor. This is going to form a complex of hormone receptor complex. Once this complex is formed, this message has been sent to the inside of the cell. The second message is this message is the which is responsible for the biological activity in the cell. So first messenger contact with the receptor. It's the second messenger. Then finally the biological activity is now we have to study something hormone signaling. Chemical signaling of hormones is basically three ways. One is endocrine, paracrine, and autocrine. Endocrine pathways, paracrine pathways, autocrine pathways. What is endocrine? Imagine this is a cell. Right? What the cell is shown here? This cell is secreting a hormone. This hormone is secreted into the blood. As the blood flows to every organ, there is a target cell which is a distinct organ. Right? So this hormone secreted from one place, secreted from the taken cell, and finally this is good. What is paracrine signaling? The word itself is really good. Right? Just next to this cell is secreting this messenger. It reaches through the interstitial fluid and then it acts on the target cell, which is very next to it. This is called as paracrine signaling. As autocrine signaling. This is autocrine self-regulating. So self-regulating. This cell is secreting which acts on the same cell to regulate its cell. This is called as autocrine. All these are called as intercellular communications. You can see this slide. There are different communications in addition to what I have just told. there are direct communication. There are the gap junctions with the sex. This is the direct communication. We also have human communication via the synaptic synaptic transmissions. Then we have the brain communication, circulating direct brain communication, the locally released hormone from neighboring endocrine cells. This one, auto-brain communication is on the hormone secreted things. Also, we have sex content communication, which good factors expressed on the cells and also we have as auto-brain communication. The vehicles in these are the impersonal communication. Now, feedback mechanisms. I think like you would have read about feedback mechanisms, but we will speak some examples related to endocrine. We all know that basically we have positive feedback. Now, this is the first example. For example, this is the negative feedback input. The condition increased with the glucose. Number one, we are consuming some food. What happens? This will increase our blood glucose level. This increase in blood glucose level will sense by the program. This acts as a sensor. Once there is an increase in blood sugar, there is going to be increased circulation. So this insulin is going to increase the glucose uptake in the tissues. That is the action. So this is the glucose uptake in the muscles, liver, etc. This glucose, as the glucose uptake increases in both tissues, the blood sugar levels decrease in the bloodstream. So this is the blood sugar. Now, because of this, the pancreas is going to again sense this that now blood sugar levels are now this will increase the insulin secretion. Because insulin secretion less now, we are back to the normal state. So this is how this negative feedback loop is operating and condition glucose levels. We'll add upon something hard. What happens during fasting? Say, for example, someone is fasting. What will happen to the blood glucose level? Naturally, the blood glucose level will fall. As this works, this will be again changed by the brain. The endocrine pancreas then separates the hormone as glucagon. This glucagon is going to increase the glucose release from as there's going to be more glucose released from the liver in the skin. The blood glucose levels will go. As the blood glucose levels increase, this is going to increase. This will increase glucagon secretion. Now, as glucagon has complex form, this how the secretion is. Now we have got the compound. This is the negative feedback. Increased blood glucose levels at the constant state. Now, before we go on to the positive feedback, since I want to know what are foods to the brain system. Say, for example, general scheme of hormones. Initially, we have the hypothalamus, which is secreting the hormones. Hypothalamus hormones. Hypothalamus will these hormones will go from the respective target glands. They secrete a very specific hormone related to that. So that is the target and this target gland. So that is a physiological. Say, for example, we will take an example of cortisol. Now, hypothalamus will be secreting cortisol releasing hormone. This cortisol releasing hormone, hypotropin releasing hormone, will be acting on and it will secrete as hormone. That's the basic. Remove from with hormone. Now, this hormone comes from secreted from will act from the renal cortex. target from the adrenal cortex, the glucocorticoid, that is the cortisol, cortisol function. So this is a general statement which I have given one cortisol. What are these groups here? As you can see in this picture, the first one we are going to deal about is long group. The example I gave you, the amount of cortisol levels will regulate the amount of secretion of particularly reason. So for long group is something like the final end product is target gland are regulating the secretion of the organs, the hypothalamic hormone. That is the long group. And so it is. Now you can understand what is the meaning of a short. A short group is something like the is necessarily controlling regulating the amount of secretion of particular protein reduced this like self-regulation. Basically, it's itself. Right? What is a positive feedback? I will say the classical example. If you be all of us would be aware of postulation, everybody up to. So the stimulus for the behavior, the head of the baby pushes against the cervix. Now, as the head of the baby pushes against the cervix, the nerve impulses from the cervix are transmitted to the brain. Now, as this nerve impulses travel to the brain signals to the pituitary gland, you simply oxytocin is that oxytocin is supposed to be a behavior. So, from the posterior oxytocin is simply. And this oxytocin is carried in the bloodstream to the target organ in the breast. You press this oxytocin. Uterine contractions, which is the AB towards the cervix. As you can see, we have more different contractions. Okay, because of this, the head of the baby is going to get pushed against the cervix. Because of this, there's going to be more impulses traveling, more secretion of oxytocin. Different contraction. So, as long as the baby is getting this process is continue. Once the baby is fully developed, then there is no push against the cervix. These impulses will be increased. Then there is increased secretion of oxytocin. Different contractions. That is how it is positive. We look about the other areas and the basic introduction. All the hormones available from each of the anterior. We have both thyroid stimulating, hormone because this follicle stimulating hormone, luteinizing hormone. All these six hormones are separated. From the posterior, hormones and B diuretic hormone, ADH hormone. The second hormone is oxytocin. Okay? These are the pituitary hormones. From the thyroid gland, we have T3, T4, thyroxine, T4, hyaluronic acid, calcium, calcium. From the parathyroid, parathormone, hormone. The endocrine transcripts from the islets of one. Here is the second glucagon. So first and we have the from the adrenal cortex, as it is divided into different layers, mineralocorticoids, glucocorticoids are 6. So from the mineralocorticoids are highest in the opposite corticoid. Glucocorticoids, they call as cortisol particles. And also we have 6 hormones. The next one will be renal. We know that catecholamines, adrenaline, epinephrine, noradrenaline, From the pineal gland, we know that melatonin is secreted, which is responsible for the thymus secretes adenosine, hymen, and some of the hormones which are secreted from here, erythropoietin, thrombopoietin, renin, 125 dihydroxyfolic acid, that is the classifier prostaglandins. And ATI natriuretic profile, brain natriuretic profile, C-type natriuretic profile are secreted. And placenta also secreted lot of hormones. Few among them are human chorionic gonadotropin, that is the HC. Human chorionic so amyotropin, estrogen, and progesterone. These are the list of hormones which are secreted from different brain hormones. Also, we have so many local hormones which will be dealing in different parts of the entire physiology. Prostaglandins, thromboxanes, prostacyclins, leukotrienes, epoxins, acetylcholine, serotonins, histamine, substance, heparin, bradykinin, a lot of gastrointestinal. We move to the next part of plasma receptors. The receptor may be present on either the surface of the cell, the cytoplasm of the cell, or the nucleus. So I am going to give you an overall view of how these receptors are present. As you can see in this picture, there is a cell here and there are various receptors. First one will be dealing with this called as membrane receptors. The second one will be cytoplasmic receptors. The third one will be here. Look closely here. The membrane receptors are named because these receptors are present on the cell membrane. So we are called as membrane receptors. Why these are called as cytoplasmic receptors? These receptors are present in the cytoplasm. And also we have receptors in the nucleus which are called as nuclear receptors. We, from this slide, just we should understand that different hormones act by different receptors. For example, the protein hormones in the catecholamines act via the membrane receptors, then through the second messenger system and activates the enzymes. The thyroid hormone acts via the nuclear receptors of mRNA in the protein synthesis. And the steroid hormones act via the cytoplasmic receptors. So remember, brain receptors, nuclear receptors, and cytoplasmic receptors. What basically are receptors made of? Receptors are large glycoproteins. Now, what are the steps of action happening at the receptor level in relation to the hormone receptors? Right. The first one will be the binding of the hormone to the receptor. So this hormone is going to come and bind to the receptor. Now, this forms a HR complex. What is the hormone receptor complex? Then the third step will be the internalization of hormone receptor complex by cytosis. Finally, we have degradation of hormone receptor complex by the lysosomal enzymes inside the cell. This is what is happening. Binding of hormones with the receptor, formation of hormone receptor complex, internalization of hormone receptor complex by cytosis, and degradation of hormone receptor by lysosomal enzymes inside the cell. But in addition to this, there is one more step here, which is the receptor molecules, after the function, they are going to get recycled back to the cell, which is also very. Now, we have seen about what is a receptor or how the receptor. Now, how are these receptors getting regulated? Basically, the receptor quantity and sensitivity are usually regulated by the concentration of hormones that act specifically on that set of receptors. Meaning, this regulation of receptor is mainly by the concentration of the hormone which is acting on that receptor. Based on that, we have basically upregulation of receptors, downregulation of receptors, and desensitization of receptors. What is upregulation? The name itself is giving you when the concentration of the hormone increases, the plasma for a longer period, the number of receptors for that hormone usually increases in the target tissue. Okay. Concentration of the hormone decreases in the plasma. Because of this, the number of receptors of that hormone in the target organ increases. This is upregulation. Also, the sensitivity of the receptor to that hormone increases. Both of these are as upregulation. The classical example is estrogen and its receptor. If you remember, we have studied the concept physiological basis of innervation hypersensitivity in previous chapters. Right? That is how the upregulation mechanism is. Physiological basis of innervation hypersensitivity. Please go back and recall about this process, how this is actually regulation now. The pictorial representation of the same thing. You can see that the cell is, this cell is having receptors in one. As you can see here, 2, 3, the receptors are getting upregulated because of the decrease in hormone. Now we move on to what is called as downregulation. The opposite of upregulation. When a hormone is present in excess, the blood for a longer period of time, the number of receptors of that target tissue is decreasing. This is called as downregulation. The classical example is insulin and its receptors. Now a pictorial representation of downregulation. You can see a lot of receptors here. As we go from 1 to 3, there's going to be decrease in the number of receptors. Down regulation. Then we have something called as desensitization. What is desensitization? When cells are chronically exposed to excess concentration, they become less responsive to subsequent exposures. This process is called as desensitization. This desensitization process you'll be reading across so many aspects in physiology. So here, with respect to endocrine physiology, they have been exposed to excess concentration. The cells are exposed to excess concentration. They become very less responsive to those exposures. This process is desensitization. What is actually happening? The sensitivity of the receptors for those hormones are getting decreased. Now, the next part we are going to see about is something called as G proteins. What are G proteins? G proteins are nucleotide binding proteins. These are actually membrane proteins which are situated on the inner surface of the cell membrane. They are situated on the inner surface of the cell membrane. These proteins are nucleotide binding proteins. These proteins play, why are we reading about these proteins? These proteins are the ones which play a very important role in the formation of cyclic AMPs. To know more about G protein, we should know something on GPC. What are GPC? G protein coupled receptor. In short, we call it as GPC. These G protein coupled receptors are actually 7 helix receptors. We call it as serpentine receptors because they take the form of a spring. 7 helix receptors on the cell membrane attached to the G protein. You can look at this picture, right? This 7 helix receptor is actually the G protein coupled receptor, which is attached to the G protein. You can see that this G protein is having an alpha subunit, beta subunit, and gamma subunit. And this bigger alpha subunit is attached to the GDP. GDP usually attaches to the we move on to the very important area, mechanism of action. G proteins. When the hormone receptor complex into these GPs, GDP is exchanged for GDP. Alpha subunit is separated from the beta and the gamma subunits of the G protein. Right? We will discuss in detail in the coming slides. The picture of the special here. You can see that this is the hormone, this is the receptor. We have the cell membrane, extracellular fluid, intracellular fluid. Also, in the inner surface of the cell membrane, we have the G protein having alpha subunit, beta subunit, gamma subunit, and our GDP is attached to the alpha subunit of the G protein. Right? This is the normal state. So, what happens next? The hormone receptor complex is formed. Once this complex is formed, we know that there's going to be a conformational change. Because of this, GTP is converted to GTP. This reaction causes the splitting of alpha subunit from the beta and the gamma subunit. This reaction causes the signal transduction and finally the physiological. This is the overview of mechanism of hormone action. G-protein, right? And the most important thing here is the splitting of this alpha subunit from the beta gamma subunit because of the right. What are the types of depending on the alpha subunit? We basically have three types. The first one is called as GS. That is alpha S. S stands for stimulation. Stimulates this alpha S subunit stimulates the enzyme adenine cyclase, which increases the cyclic AMP formation. That is why it is given as S. We have another G protein called I, in which the alpha subunit is alpha I. The I stands for inhibition. So this inhibits the enzyme adenine cyclase, which decreases the cyclic AMP formation. In addition to these two, we also have Q protein, that is the alpha Q subunit, which activates the membrane enzyme phospholipases. Right? So we have three types of G proteins, ES, EI, and EQ, which are having alpha S for stimulation, alpha I for inhibition, and Q for activation of membrane enzyme phospholipases. Right. What are all the hormones which are acting through the CPC? This list is very important. As you can see, catecholamines, acetylcholine, histamine, serotonin, adenosine, angiotensin II, ADH, oxytocin, TSH, FSH, LH. All these hormones are acting on the. Now, we come to the classification. Hormones are basically classified into three types. One is peptides, amino acids, and steroids. What are the classification of hormones? Under the peptide hormones, we have different families. We have the insulin family, which has insulin-like growth factors, and we also have the glycoprotein family, which is the LH, FSH, TSH, and HCGs. We have the growth hormone, lactate, human placental lactogen. All these comes under the growth hormone family under the peptide hormones. Also, we have secretin family, which are secretin, glucagon, VIP, and GLP. And in addition to these, we have calcitonin, AMP, cholecystokinin, ADH, inhibin, somatostat, ACTH, also comes under the peptide hormones. The next comes the amino acid derivatives. This is the second type. These are amines. Epinephrine, serotonin. All these are there. We also have iodinated amino acid, that is the E3 and E4. Right? These are all amino acid derivatives. Now we have the third group, that is the steroid hormones. Those are glucocorticoids, melanocorticoids, estrogen, progesterone, testosterone, 125 dihydroxyproline. So basically, you have to understand there are peptide hormones, there are amino acid derivatives, and there are steroid hormones. And these are all like hybrid topics for MCQs. They can ask you, melanocorticoid comes under which classification of hormones? They can give you options. Those options. Okay? So try to memorize all these things. Now, we come to the next stage on second sequence. As the name suggests, I think in the second slide or the third slide, I showed you a picture on first messenger, second. The same continuation. The intracellular signal that formed by a series of enzymatic reactions subsequent to the formation of hormone receptor complex or the signal as second messenger. So, actually, the hormone is the first messenger, and after the formation of hormone receptor complex, there's going to be an intracellular signal molecule that is formed by a series of enzymatic reactions. These are called as second messengers. Now, what are the second messengers we know? The most important ones I have said. We have the cyclic AMP, diacylglycerol, that is DAG, inositol triphosphate, that is the IP3. We have cyclic GMP, we have phosphoproteins, we have the restricted mRNAs, also we have intracellular calcium. So, list of second messages very important. Cyclic AMP, DAG, IP3, cyclic GMP, phosphoproteins, transcripted mRNAs. So, intracellular calcium. And we have second. Now, we come to the important stage of our class today, that is the mechanism of protein hormones and chemicals. Okay. If you remember, these are actually acting on the brain receptors. Now look at this picture. Now, this stage one, right? This stage one is the resting state. This is the hormone. As you can see, this is the cell membrane, this cytoplasm, and we have the receptor here. We have the G protein, alpha, beta, gamma subunit, to which alpha subunit GDP is in the state that is called stage one. What happens? As the hormone is coming in contact with the receptor, hormone receptor complexes. As this hormone receptor complex is formed, we know that there is going to be a confirmation from, as you can see in this picture, the GDP in this is attached to alpha subunit, the normal state. After this hormone receptor complex, this GDP is converted to GDP, and the alpha subunit has been detached from this beta and gamma subunit. So this is what is happening once the hormone receptor complex is formed. There will be a conformational change. I think I have explained this in the previous slides. So this is happening in the system. And because of this, there is going to be activation of adenyl cyclase. Activated adenyl cyclase will turn ATPs to cyclic AMP, which again on activation will activate the protein kinases A. Finally, this protein kinases A will act upon, and the physiological action of that will be exerted. Right? Okay, as you can see, I have put up 1, 2, 3, 4, 5, 6, and 7. So that you can remember this in a stepwise hormone receptor complex is formed. That is going to have a conformational change here, because of which there is going to be change in the GTP alpha subunit, and this is going to get detached, which activates the inactivated, previously inactivated adenyl cyclase, which again acts on the ATP to convert it to cyclic AMP. The cyclic AMP is activated. This activates the protein kinase A, and finally there is a response to it. Now, once this hormone has finished its function, all these processes are done. Now, what is happening at the next level? Now, this GDP, what we have seen here, is having something called as the GDP is having the GTPases activity, intrinsic GTPases activity. So, once this binding process is over, what happens is this GTP, because of its GTPases intrinsic activity, is again converted to GDP. As this is converted into GDP, the complex which has been split, this will go and attach the alpha subunit will go and now attach beta and gamma subunit from the previous resting state complex of alpha, beta, and gamma. This is how restoration is established. Okay. This is the mechanism of action of protein molecules. How is the mechanism of action of steroid? Look at this picture. You can remember the steroid hormones are going to act via the cytoplasmic receptors. So again, we'll start with the same process. This is how I'm going to explain all the mechanism of hormone action. 1, 2, 3, 4, 5. So it is easy for you to follow. The first step is the hormone receptor complex. The hormone receptor complex is formed. The hormone receptor complex is going to then activate the DNA processes, then the mRNA process, and we all know that translation transcription processes are going to be activated. Then we have the mRNA formation. The mRNA moves out of the nucleus. Then it is going to activate the ribosomes to form proteins, and finally we have a response of this. So what is our basic understanding of the steroid hormone mechanism of action is the protein hormones were membrane receptors, but here these are receptors in the cytoplasm. Then this hormone receptor complex is going to activate the DNA binding sites, and DNA activation will cause like mRNA transcription processes get triggered. Then we have the mRNA. Then it goes out, ribosome will go that way. Then the response. So this is how steroid hormones are. The second messengers which I was talking about are mainly formed by activation of four main transcription systems. We are going to deal with it little more detail. The first one, adenylyl cyclase, adenylyl cyclase, cyclic AMP. The second one will be membrane phospholipase, phospholipid system. The third one will be guanylyl cyclase, cyclic GMP system. The fourth one will be intracellular receptor systems. Right? These are the four systems. One by one, I'll just try to explain these two here. The first one will be the adenylyl cyclase, cyclic AMP system. Now we have the cell membrane, we have the ECF, this is the ICF, this is the hormone. What is happening? The hormone comes up right, the receptor, hormone receptor complex is formed. This causes or this stimulates GS or inhibits GH. So this is the next action. Anyone can do that, right? Either GS or GH, depending on the alpha subunit. This will activate the adenylyl cyclase. Now this adenylyl cyclase activation will cause ATP to will change ATP to cyclic AM. In cyclic AM, this can degrade to 5-AMP by phosphodiesterase and it can be recycled. What is the main function of this cyclic AMP? Is this cyclic AMP converts this inactive protein kinases to active protein kinases. This is the next important step. Then this activated protein kinases A will result in phosphorylation of many cellular enzymes. This will help in the synthesis of phosphoproteins. Finally, this will exert the physiology. In addition to this, right, what is happening here is this activated protein kinase, this moves into the nucleus and the box design showing here is happening. Right? So this activated protein kinase moves into the nucleus and it helps in the phosphorylation. PRPs, that is the cyclic AMP response element binding proteins. This will help some transcription of genes and the protein synthesis. This is the overall action of the adenylyl cyclase pathway. Right? Adenylyl cyclase, cyclic AMP pathway. So, more time I will explain. First of this hormone receptor complex. This will activate the adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic AMP. This cyclic AMP converts the inactive protein kinases A to active protein kinases, which again helps with phosphorylation of cellular enzymes, synthesis of proteins, finally expression of its. In addition to this action in the cytoplasm, it moves into the nucleus, where this activated protein kinase A will help in phosphorylation of CRPP. That is cyclic AMP responsive element binding proteins, which helps in transport from genes and finally the protein synthesis. We move on to the next one. Membrane phospholipase, phospholipid system. Membrane phospholipase, phospholipid system. Yes, the same kind of a picture. We are going to start with hormone receptor complex formation. Same picture. This is the cell membrane, the ECF, hormones, and receptor. The hormone receptor complex is formed in the first step. This is going to activate the GQ protein, G protein. So, in the types of G protein, these are GS, I, G. This is activation of GQ protein, which is going to activate the phospholipase C. This activation of phospholipase C will result in formation of PIP2, that is phosphatidyl inositol bisphosphates. This activated PIP2 will then split into two pathways. One will be your IP3 pathway, there will be your DHG pathway. That is your inositol triphosphate pathway and also your diacylglycerol pathway. Now, what is happening? We just see this slide. The IP3 pathway, this is going to stimulate the endoplasmic reticulum to release more of calcium. This calcium is going to bind with calcium binding protein and the other things. Physiological efforts. On the other side, this DAG is going to activate the protein kinase. Activation of protein kinase C will result in phosphorylation of proteins. Phosphoproteins are formed, and finally they are going to happen physiological. Right? This is how the membrane phospholipid system works. In repeat, hormone receptor formation, activation of GQ, activation of phospholipase C, activation of PIP2, that is phosphatidyl inositol bisphosphate. This gets us two pathways, IP3 pathway and DHG pathway. This IP3 pathway is the activation of endoplasmic reticulum to release more amount of calcium, which binds with calcium binding protein. Physiological effects of it will be significant. On the other side, DAG will activate the protein kinase C, which activate the phosphorylation of intracellular proteins, formation of phosphoproteins are formed, then finally these results in physiological. The third one will be guanylyl cyclic GMP system. What is happening here? The same process, everything is the same here, but there's a slight change. The hormone receptor complex is formed. Once this receptor is formed, you can see that this is going to activate the guanylyl cyclase. This guanylyl cyclase helps in conversion of GP to cyclic GMP. This cyclic GMP is going to be the one which is going to open the ion channels for inward movement of the cyclic channels. Now, this cyclic GMP will activate the cyclic GMP dependent kinases and phosphatases. So, this activation will result in phosphorylation of intracellular proteins. Same like the last time, phosphoproteins are formed and they are going to exert their physiological effects. I'll just try to explain one more time. Hormone receptor complex is formed. This time, this is going to activate the guanylyl cyclase. Because of this, GP is getting converted to cyclic GMP. This cyclic GMP is an activation. Cyclic GMP dependent kinases, phosphatases, which ultimately helps in phosphorylation of intracellular proteins and formation of phosphoproteins, and they are going to exert their physiological effects. The fourth one is the intracellular receptor systems, which is the transcription. Right? So what is actually happening here? You can see in this picture, the hormone is directly going into the cytoplasm, not like the previous ones. Right? Now this is going to the cytoplasm. Now from there, it is going to go in, attach with the receptors in the nucleus. Actually, what happens is the DNA binding site will not be exposed. That will be protected by something called as HS, that is the heat shock protein. This heat shock protein will cover the DNA binding site. Now, with the hormone receptor complex formation, conformational change happens because the heat shock protein gets disintegrated from this, and this exposes the DNA binding sites. Because of this, this is going to actually activate the hormone response element, that is the enhancer. These binding sites are going to activate this hormone receptor, hormone response elements, which is going to result in transcription, formation of RNAs, also finally result in protein synthesis. So, what is the difference in the other system? The other system we saw in this one is the hormones are entering into the cytoplasm and then into the nucleus, but before to the attachment of this DNA binding is covered heat shock proteins. Once these heat shock proteins, this hormone receptor complex is formed, these are getting disintegrated. This binding site, DNA binding site is exposed and the trigger for hormone response element. start the process of transcription, RNA formation, then finally protein synthesis. Finally, these will give the responses. Right? Now, at last, we come to the last few slides of our class. There are a few other mechanisms which you should know. Something on tyrosine kinase activation and also on calcium carbonate system. What is something special about tyrosine kinase activation is they will not require any glucose. They get activated by a process of autophosphorylation. Right? And then also finally we touch upon calcium carbonate system. What is this tyrosine activation? Tyrosine kinase activation. As you can see here, the hormone receptor complex is formed. As this is formed, you can see that there are phosphorylation sites present in the intracellular domain of this system. So, before the attachment of this hormone, resting state, usually these phosphorylation sites are not expressed. Hormones binding with the receptor, the intracellular domain of the receptor is an autophosphorylation site exposed. Because of this, the tyrosine kinases this acts as a primary substrate for phosphorylation. Phosphorylation of serine pionine kinases. This will activate the enzyme, also help with gene transcription. Finally, that's the physiological. We need 1, 2, 3, 4 for inflammation. Hormone receptor complex is formed. This exposes the autophosphorylation sites, formation of tyrosine kinase or activation of tyrosine kinase, substrate phosphorylation. Because of this, phosphorylation of serine kinase is complex. This activates the enzyme in transcription, because of which the physiological effects are discovered. Okay. The last one that we are going to see today is calcium calmodulin system. What is this calcium calmodulin system? You can see here, again the same process of hormone receptor complex. This hormone receptor complex formation will stimulate the ligand-gated calcium channels too. Because of this, there is going to be influx of calcium into the cell. Now, this influx of calcium into the cell will increase the cytosolic calcium level. This increased cytosolic calcium levels can just get exchanged with 2H plus and also 3Na plus through the calcium H plus ATPase. Right? In addition to this, increased cytosolic calcium levels will activate another set of processes. As you can see here, mitochondria and endoplasm. This increased cytosolic calcium level will also trigger these mitochondria and endoplasm to secrete more amount of calcium, to release more amount of calcium. And this will activate the IP3 pathway. Right? We have to remember the IP3 pathway which I was talking there. This helps in influx of more amount of calcium, store operated calcium channels. Now, what is actually effective is, as there's going to be increased cytosolic calcium levels, calcium are also released. Now, the store of this, the calcium store of mitochondria and endoplasm and reticulum are increased. To replenish this store, we have a store operated calcium channel, which is now because of this process. This helps in influx of calcium to this endoplasmic reticulum and mitochondria, which replenishes this calcium. Right? And in addition to this, all these increased cytosolic calcium levels will bind to the calcium binding proteins and their physiological effects. Right? So I think that finishes off the entire class of small introduction of all the endocrine glands, various hormones secreted, different receptors on this. Then we touched upon the different pathways of how the membrane receptor acts, how protein hormones, steroid hormones, how the cytoplasm. And also we saw various systems of cyclic A, B, adenine cyclic, cyclic A, B system. We saw the phospholipase C system, we saw the cyclic CFA system, intracellular system, also the tyrosine kinase activation system, so calcium channel systems. Okay. Thank you.